one and all. I, Dr. Sakshi Bhadwaj, on behalf of Elton Technologies, extended my warmest welcome to those who have been with us since from the starting of this Women's Day special webinar series, and to those as well who are new to our this series. It's actually as appreciated effort that Elton Technologies has taken up that initiative to appreciate or honor the contribution and appreciation of all the women researchers in their respective fields. There is one saying actually, when you gravitate too much on your results, okay, you go through the cycles of frustrations and fear. But when you gravitate more on your results or your efforts, then you go through the cycles of excitement and success, right? So, and we all heard about that malaria, right? Basically, it's caused by the Plasmodium Falciparum, which is a parasite. And this parasite is transmitted through the infective bite of, Anopheles mosquito. This parasite is fatal among all the other strains of strains belonging to this particular family. There is a very interesting story behind this name malaria actually. In 18th century, this malaria name actually this indicate bad air. In 18th century, the people used have that hypothesis that this malaria caused by taking the breath into the bad air in the marshy areas. But in 1880, those scientists had disproven this theory behind this malaria, spreading one. So, but there are some medication available into the market for malaria. However, our researchers continue to investigate the different targets and the therapeutic candidates to ease this global burden. We have such a researcher with us today for this masterclass, which is entitled as In Silico Evaluation of Inhibition of Plasmodium Falciparum, a conjunction of structure-based approach with structure prediction and molecular dynamic simulations, where our expert will be discussing about a systematic biological approach along with a structure-based pharmacophore method to inhibit the target. I am extremely overwhelmed to introduce our special guest for today's session, Dr. Sanchaita Rajkhowa. She is computational researcher working as an assistant professor in the Center of Biotechnology and Bioinformatics, Dibrugarh University, Assam. She has completed her MSc in the Bioinformatics from the Dibrugarh University in 2010 and obtained her PhD degree from the Tezpur University, Assam in 2017. She has an expertise in the molecular modeling and computational biology with the different molecular modeling tools. Computer-aided drug designing is the primary focus for her studies. She has published her work in numerous reputable national and international research journals like Elsewhere, Ville, Royal Society of Chemistry, etc. are the name of few basically. Now, without taking too much time, let's start. It's up to you, ma'am.
Thank you, Dr. Sakshi, for the beautiful introduction. It's really overwhelming to be presenting to this audience today. So, good morning, everyone. I am Dr. Sanchaita Rajkhowa, like Dr. Sakshi introduced me. I have been working in the field of computer aided drug design since 2011. And I have joined the Dibrugarh University in 2019. So currently, I am engaged in both teaching as well as research. We do have a dry lab facility in our center, as well as a wet lab facility wherein we perform the antimicrobial tests, cytotoxicity tests, and also we do have a dedicated cell culture laboratory. And our today's my talk is basically on the computational approach, and it is involves the complete dry lab, and it mostly concerns with the inhibition of malaria. as the initiative taken by the government of India to completely eradicate malaria by 2030. 2030. So, like she said, malaria, also known it was known as malaria. That means bad air. But now we have, with gradual study, it has been proven that it is not actually the air. It is the falciparum, the mosquito, that is responsible. My study has the following contents. Malaria needs no introduction. It is one of the most life threatening diseases till date, and it has been causing havoc since a very, very long time. It is initiated by the Coccidian protozoa parasite and infects the circulatory system of humans. The parasites are transmitted to person through the bite of Anopheles mosquitoes, and they are commonly called as malaria vectors. They are mostly widespread in tropical and subtropical regions and are caused mostly by five species of the parasite. Out of which the falciparum is the most deadly and lethal. And of course, Vivex also possesses a threat. It spreads, it's very widespread, but it is comparatively less dangerous. Now, this is the epidemiological trend that you can see of malaria in India from 2000 to 2019. As you can see that it has been gradually decreasing. But there was a sudden rise in this is the data that I have given here is from the World Health Malaria Report of 2022, which says that in 2021, malaria, an estimated amount of 247 million cases arose in 48 endemic countries. This was an increase from the previous statistics of 245, which is an alarming concern. So, in the last 2 decades, this malaria, this falciparum has achieved or has become resistant to almost all the anti-malarial drugs. Okay, these resistances mainly occur due to pan-point mutations. Now, most of this research that has been happening in the recent years are done towards inhibition or emergence of drug resistance. But there is a need of new drug, and towards which few efforts have been done in developing of drug of new mechanism. That means of finding out of a new target. Malaria, it is addressed as actually a poor man's disease. So a lot of the pharmaceutical companies, they also do not like to invest in poor man's diseases. That is another problem. So this study was designed to find out a new target maybe, and also the ways of inhibiting this new target. So with this objective, with this particular problem in mind, we laid the objective to study the various metabolic pathways of the Plasmodium falciparum, then analysis of all the protein-protein interaction networks, and maybe establish a new drug target. Then we also tried to model the protein as the crystal structure of the protein that we found as the good, ideal target was not available. Therefore, we tried to elucidate the structure. Then we performed the structure-based pharmacophore and generated a pharmacophore based on the structure of the target. And then we used that pharmacophore to map onto various libraries and then come up with new potential compounds. So these are the methods and tools that we used. We used the R programming language for text mining. Then we used the Plasmodb database to get the pathways, to study the pathways. Then we used the Cell Designer 4.4.2 to design the metabolic pathway. And we used the string database to version 11 to get the protein-protein interactions of that particular metabolic pathway. Finally, we analyzed the protein-protein interactions using Cytoscape. 3.8.0, and we used Discovery Studios to model that protein. Now, this method you can see is divided into two parts, that is the green and the yellow part. The green part is mainly for the protein part, okay, the target part. And the yellow part consists of the small molecule, the ligand or the screening, you say. So once we got the structure, we used that structure. And first we did is build the library. We built the active library by mining inhibitors from the Campbell as well as the binding DB database. And for the inactive molecules, we generated the decoy library from the DoD database. Then we used this both active and inactive ligands, and we generated the pharmacophore using Discovery Studios. We used that particular pharmacophore to screen libraries. The library consisted of the FDA approved drugs from Drug Bank and also the natural compounds from the IBS database. And then we used numerous virtual screening techniques implemented from Discovery Studios, and we found out three compounds. For the text mining, now the information search and extraction from articles is very time consuming. Okay, so text mining, what it does is it speeds up the process of searching and extracting specific data. So using the PubMed database, the malarial associated genes were abstracts were collected using the R program. We used the PubMed.mineR module for this purpose. The gene list retrieved by text mining extracted a total of 448 genes. But when we analyzed this gene list, most of the genes were either well-established target or not so crucial, were found not so crucial. Therefore, again, we performed a manual literature search. And we found that the plasmapsin 5, this is a very important protein for the survival of the Plasmodium falciparum. Okay, what it does, it exports the protein by cleaving the pexyl protein. So therefore, taking this particular PMV, the plasmodial PMV in mind, we proceeded with our further analysis. Now, we considered for our study, we considered 6 different pathways from the PlasmoDB database. But we found that 2 of them, that is this phosphoinositides and membrane traffic, as well as the post-translational translocation in eukaryotes, these 2 pathways were essential for the survival of the Plasmodium falciparum. Now, although every pathway has been reported in the involvement of endoplasmic reticulum activity, still there was very little, there was no clear evidence of this plasmapsin 5 being involved in any other 4 pathways. Okay, so also there were 6 more pathways. They were based on computationally inferred by the orthology, but we couldn't find the EC numbers. Then we performed, we took these pathways, we tried to design these pathways using the cell designer. This is the phosphoinositide and membrane traffic pathway. So we visualized and analyzed. Now to visualize and analyze these these two metabolic pathways are constructed in cell designer. This is the first one, and the next slide you will see the next one. The selected pathways were modeled and visualized using the cell designer version 4.4.2, by the way. The pathways are based on availability. This is purely on the availability of literature and data in the PlasmoDB. The phosphoinositide and membrane traffic pathway is verified with the available pathway of Plasmodium falciparum in the KEGG database. We have verified this pathway from the KEGG database, but the PMV activation was not available through the pathway. It's not available. But then we know that it is the same. Okay, and the post translational translocation in eukaryotes pathway was adopted. The next one, that it was adopted from a work which was done experimentally by a group. Okay, she was Marapana and her group, they did it. So we used that particular pathway to model our post translational and translocation pathway. This pathway was discovered as precursors for specific second messengers that are involved in signal transduction, but now it acts as a center stage in controlling most of the essential processes at every cell membrane. Thus, this phosphoinositide, they regulate the membrane dynamics and the vesicular transport. On the other hand, the Plasmodium falciparum exports remodeled proteins by cleavage of a pentameric motif, okay, at the N terminus by this plasmapsin 5. It is located in the endoplasmic reticulum. This PMV, it interacts with other proteins through which they recognize the effector protein for the protein export. This whole pathway, it was designed by taking into consideration the work of experimental work of the one that I spoken before. No, she was. I think yes, she was Marapana. So from her work, we designed this particular pathway. Then what we did is we selected the pathways, protein-protein interaction network from the string version 11.0, and then we constructed this, we merged both the pathways and we constructed the networks in cytoscape. Then both the PPIs were subjected to topological analysis, and we also applied the CytoHubba plugin for identifying the central node. To establish the importance of this PMV, we performed the static knockout. The PPIs for both these pathways were extracted with having an interaction score of 0.007 confidence from the string database. And this PPI is constructed, and this one, the one that you can see, it is constructed by merging both the pathways that we discussed in our earlier slides. Then the network analyzer was run for having a better knowledge. This particular this particular network that you can see consists of almost 124 proteins with 597 interactions. This is the result of the CytoHubba plugin, and you can see that the global based methods, they ranked this PMV as the essential protein. These are the visualizations of the other global parameters like the betweenness centrality, the closeness centrality, wherein you can see that the PMV, it is occupying a very good position. Then we performed the static knockout study. Knocking out an essential protein, it results in lesser interaction of the protein. Okay, so which leads to the loss of certain essential functions in the network. So if you knock out one protein, you can find out what essential function it is losing, right? So a comparison study was conducted between these two networks by removing this PMV. First we did is, this is the merged overall topology of the merged PPIs. Then we try to remove the PMV, and then we see, analyze the results, and then we try to randomly remove two other proteins, keeping this PMV and randomly removing two other proteins, and then we try to analyze. The removal of PMV in this number B, okay, it results in increase of clustering coefficient 2 0.700, as you can see. The shortest parts were dropped to 49%, while the removal of the two random proteins in number C showed decrease in the clustering coefficient, but there was no decrease in the shortest paths percentage. So summarizing, all these results and also of the global parameters, this PMV was identified as an essential protein, and we came to know that it plays a crucial function in the network. Therefore, there is a possibility that if we target this PMV, it may lead to the eradication of this Plasmodium falciparum altogether. Then, now the problem was that the crystal structure of this Plasmodium falciparum PMV is not available till date. So we tried to model it. To model it, we retrieved the primary sequence of the target PMV, Plasmodium falciparum PMV, from the UniProt KB database. Okay. Now, since this PFPMV is an essential membrane protein, okay, but its active site doesn't fall inside the membrane, but it is an essential membrane protein, and it's N terminal as well as the C terminal. So some of its amino acids, so they are embedded, partially embedded in the transmembrane part. Therefore, when we considered modeling of this protein, we actually did not involve these the residues belonging to both the C and the N terminal, which were partially embedded in the transmembrane part. Then we considered the other residues, and we performed a blast to find the template structure of known proteins from the PDB database, and we found that this 4ZL4, that is the plasmapsin 5 of Plasmodium vivax, okay, this was bound to a transition state mimetic called the WEHI 842. This was identified as the best hit, and it showed an identity of 63.83%. Then we considered both the 4ZL4 and our Plasmodium falciparum PMV5, and we performed a pairwise alignment in using the emboss needle, and we found that the entire stretch was not aligning. In fact, from this, you see the residue G72 to arginine, glycine 72 arginine 582, if you can see, only these residues were aligning, and this particular alignment was, there was a query coverage of 80% similarity. Therefore, we considered this, but there is a stretch of missing residue. Can you see here? There is a stretch of missing residue. So this stretch also we tried to model. I will tell you how we did it. So for now, you have to remember that this glycine 772, for our study, we were considering as a glycine 1, residue 1, because the other residues we did not consider as they were embedded inside the transmembrane. Similarly, from this particular residue, that is 529 onwards, these were also embedded inside the transmembrane. Then we also performed the multiple sequence alignment of all the plasmapsins that were present, and we found these results. And this was also done using the Discovery Studios. We performed the homology modeling of the protein using the Discovery Studios. We used a Ramachandran plot to analyze the phi-psi torsion angles, and that we performed using the Rampage web tool, and the PROSA web portal was used to analyze the Z-score of the 3D model, and the best model was with the most negative dope score, and Z-score was selected. Now, according to the table, as you can see, this model 1, can you see it has the lowest dope score? and as compared to all other four models. And the RMSD also was very small as compared to the other models. And the correct fold estimation is a crucial step in model determination. So the Z score, what is the Z score? The Z score helps in finding the fold confidence by showing the highest identity with the template sequences. Higher the value of Z score from POPROSA survey, you can see the better the fold confidence. So here also you can see the model 1 was showing the best highest Z score. Also we seen the Ramachandran plot analysis, and here also we see that only for one residue was lying as an outlier and all other were in the favored region. Therefore we consider this first model out of all the more than 20 models that were generated from the Discovery Studios. Then, as you remember that I told you there was a stretch, this cyan box, if you can see in the alignment on the towards the right, the cyan box, that was the missing residue. So what we did is for our study we used a Galaxy web server for the refinement of the conformation of this particular insert 2 region. We are calling it as the insert 2 region. So this missing residue was modeled using the Galaxy web server. The structure of the modeled the Plasmodium Falciparum PMV was found to have a very typical aspartyl protease fold, having a crescent shape and a predominantly this beta sheet. And this is just like the core. This core is just like that of the Plasmodium vivax PMV. The model structure also revealed 15 cysteine amino acid residues, which I have colored as yellow. So these were found to be conserved among the other PMB autologs of the Plasmodium species. And also, these particular residues were responsible for the stability of the protein. The PFPMV, this NAP insert that you can see, the one in green in color, this is not present in other food vacuoles plasmapsins, and this forms a surface loop, the green one. Also, the beta flap, uh, which is colored orange, this part. So the beta flap is thought to play a significant role during the catalysis. The model, this particular model also has a helix turn helix, this red color. Okay, this motif is known to be conserved within the Plasmapsin 5 autologs of all species of Plasmodium. And this is also very similar to the crystal structure of the Plasmodium vivax PMV. Okay, and this structure was actually elucidated by a group, Hodder and their group. They have done this study, so this is very similar. Also, this particular model, this insert 2 that we have modeled using the Galaxy web server. So, this is showed as a cyan color. It shows a helix turn helix structural fold, which was predicted similar to one of the studies that was performed by Bedi. Okay, and also there is another group which have done a similar study using a different program known as they were the Citicul, I think, Citicul and group. They also predicted this particular insert, but it was unstructured in their study. So in our case, we have actually elucidated this part as well. Now, the structure of this template, the template was the 4ZL4, was then compared with the structure of this WEHI 482, which we docked onto the modeled PMV, that the one that we modeled. Okay, this is the crystal structure and this is the model part. The A one is the model part. The here yellow is the Wehi, the B is the crystal structure with the Wehi 842 insert. Okay, the crystal structure of PVPMV in complex with Wehi, I told you, was solved by Hodder. Okay, they found that this N-terminal benzyl carbamate moiety, there is an N-terminal benzyl carbamate moiety of Wehi 842, which is projected just outside the cleft, but it does not directly influence the binding affinity of the inhibitor. But this oxoguanidine, there is an oxoguanidine ion of Wehi which is found lying deep within the S3 pocket. Okay, and it forms multiple interactions. Now, the interactions that it forms, we actually submitted our model to the FTMap server, and the ones you can see the C and D here in the graph. So these are the green ones are the H bonds, and the orange ones are the non-bonded interactions. So these residues, they are quite common to the structure that was, similar to the one that was solved by Hodder. Then we performed the molecular dynamics of our model to optimize the structure. You can see that from the RMSD plot that the structure, the RMSD is almost stable with very little fluctuations up till 100 nanosecond. And also if you see the radius of gyration, here also you would see that the structure had shown some fluctuations up till 40 nanosecond, but post 50 nanosecond there was a tendency of it becoming achieving the compactness, the desired compactness. Therefore, then what we did is we also finally tried to superimpose the structure, the one that we got from molecular dynamics, with the crystal structure, and found that there was an RMSD of 1.09 angstrom. The proteins, they seemed well superimposed onto its template. Okay, except for the cyan part, which is the insert region, which was not solved by Hodder. And uh, and since Hodder also reported that Wehi 842 shows, a moderate activity towards the Plasmodium Falciparum PMV. So in A part, in this figure, the A, it is showing the interactions of Wehi with our template, that is the 4ZL4, and the B part is showing the interactions with the model. Since it's showing moderate interactions, so we believe the number of interactions should be less, and the C part is the superimposed part. Now, from the study, we found that the structure that we modeled is almost correct. We believe that it is correct, and using this structure, we actually went ahead with our further study. We performed the structure-based pharmacophore generation thereafter. For this, the active library was, we chose almost 61 molecules. Okay, for the active library, against plasmapsin, all the 61 molecules were plasmapsin inhibitors, and they were retrieved from plasmapsin 5. They were retrieved from binding DB database and the Campbell database with their IC50 values. Okay, and for the decoy library, the inactive library, we used the DOOT database for generation of 900 decoy against the active compounds whose IC50s were less than 100 nanomolar. Then we used this model protein as the receptor, and Wehi 842 was used as the ligand. So this is the model protein, and this is the Wehi. The figure on top, A is the model protein and B is the Wehi, and the figure just below is the defined binding region. Then we generated, the software, the Discovery Studios, we performed a receptor ligand pharmacophore, and we generated actually 10 pharmacophore hypotheses. From this 10 hypotheses, the best pharmacophore has been shown here you can see the values as well, the ROC curve of the first model, and this was found to be the best pharmacophore model. And this had three hydrophobic bonds. The one colored cyan. Also, it had two hydrogen acceptors, the ones which are colored green, and three hydrogen bond donors, which are colored as magenta. So, this is the best pharmacophore model, and the distance between the two adjacent atoms have also been shown in the figure towards the right. Though the distance between the adjacent atoms was also mapped onto the first model. Now, using this first pharmacophore model, we try to screen out our compounds. First, we did is we built a compound library of 71,606 compounds. These were from here, the 68,931 were from the IBS database, which had the natural compounds, and 2,675 compounds were from the FDA approved drugs. We wanted to see if any particular medicine could be used as a repurposed drug. So that was another motive for the study. Then we mapped our pharmacophore, generated pharmacophore, onto this particular database, and we found that of these compounds, only 24 actually matched the specifics, our generated pharmacophore. So this 24 molecule, compounds, then again we docked onto our model protein. When we docked it, we found that uh, only 13 compounds of the 24 compounds, they had acceptable C Docker energies. Then we also evaluated the binding energies, and we found that of these 13 compounds, so virtually we were screening, and we found that only 9 compounds showed good negative binding energies actually. Then also the nature of interactions we took into consideration. So the ligands interactions with the protein we checked, okay, and we tried to we tried to compare it with the previous studies, and we found that of these compounds, only three compounds, uh, uh, three compounds qualified that particular, I mean, the interaction part. These three compounds had a zinc ID. So I'm giving the names here, zinc ID with the C Docker energy and the binding energies. There is another compound which has a stock ID of the second compound, which is available in zinc database as well. You can purchase these compounds, and then you can perform, one can perform the experimental studies as well. We've also given the C Docker energy as well as the binding energies here. And for the third compound as well, we have given these are the docking results. Here you will see the 2D representation of the docked ligands, and these are the solvent accessible surface area of the screen ligands onto the model protein. Also, we tried to perform a DFT analysis to see their, electronic configuration, and we found that all of them showed similar differences of homo-lumo gap. That means the energy of, having a similar homo energy lumo energy gap. That means they are all the reactivity. Or if you consider the reactivity also, they are almost same, with the first one showing, a slightly more reactivity. Therefore, from the study, we concluded that plasmapsin 5 could be a potential inhibitor, could be used in the inhibition of maybe Plasmodium falciparum in future. Then we used that particular protein, and we performed the pharmacophore, structure based pharmacophore. We actually generated the pharmacophore, and using that pharmacophore we screened libraries both natural compounds as well as the FDA approved compounds. And we found that three, using various tools, we found that maybe three compounds, okay, which could be potential inhibitors of this target. So, this particular study can also be considered as a pipeline to perform various in silico inhibitions of maybe other possible targets. These are some of the references that I have used during the preparation of the slides. These are my students. The student on towards the right, there is this male. There's only one male actually, so there are lots of females in my group. So on towards the right, this boy, he is Abhichandran Das. So most of these works are done by him, and there is my other group. So these are the ones towards the left are the younger ones. From here, the one who is standing on my right, the girl, she is Neha Dev. So part of this work was also her contribution. So I acknowledge my team. I acknowledge Durga University for providing the lab facilities. I also acknowledge Alton Technologies to give me this platform to speak of my work. I also acknowledge Dr. Divya from Alton Technologies. She actually helped me a lot while working with the pharmacophores. So thank you, everyone.
Thank you so much, ma'am. It was very informative session, and you have well explained that how the multi-drug resistance is causing the issues while taking one drug candidate, and how it won't work on the disease condition, which is now extended to the XDR, that is extensive drug resistance, and now it is like total drug resistance, means no drug can
Exactly.
bind and show the significance activity basically. So you have explained how starting from the identification of how the homology modeling involves in detection of a particular 3D structure of a protein, and how we can utilize that 3D structure of that particular protein to for our drug discovery process. We have some questions, so we'll take those one.
Yes, please.
Can you please explain importance of shortest path?
Yes, so I think yes, you are talking about the shortest path in case of the our knockout process. So whenever you are performing a knockout, so when you try to compare between, so whenever you find the shortest path, okay, when you see the shortest path, then you will see that it is. I mean, if the path becomes shorter, that means there is certain significance of the protein, right? If you knock it out.
Okay. Next question. Importance of DFT calculation in the drug discovery.
Yes, so here we DFT, and we apply DFT to see that sometimes we see the energies of both the homo as well as the lunar. So we try to see how much reactive the molecule is. So you see, there is a higher energy and a lower energy, isn't it? So a molecule, the energy has to jump from a higher and a lower energy. So we always try to see if it has what is the difference, how much time the energy, obviously if it takes some more time to jump from a lower to a higher orbital, that means its reactivity will also be affected. I hope I am answering it.
Yeah, basically, actually, add on to ma'am, this DFT values we are calculating with the help of Discovery Studio as a
Yes.
density functional theory.
Functional.
That is basically for the optimization of your geometry of your
Mhm.
protein or the target
Mhm.
molecules. So
Yes.
yes, next question. Okay, what is the significance of RMSD values in the selection of protein structure?
Yes, so you see, RMSD we try to superimpose, isn't it, with a known structure. If the structure actually varies too much, then we get a higher value of RMSD. But since there was in our case, we had a 80% similarity. So we got a good RMSD. That means if the RMSD is lower, your structure will also be, you can say it is better in the sense that it is not deviating too much from the template.
Yes, correctly said. RMSD is basically root mean square deviation.
Yes,
Basically, we are checking the how it deviates from the one of the query structure to the your other structures
yes,
which are because we are checking the alignments of
exactly.
this thing. So even in the if I'm this in simulation dynamic studies as well, we are checking the RMSD, RMSF values. We are
Mhm.
checking the root deviation and the fluctuations of our particular complex in during by providing the what you call it as real time environment to that particular complex. So yes, that
Exactly.
is the answer. Yes, thank you. I think we have that much question. Yeah, thank you so much, ma'am. It was very informative session actually. How because it's these conditions, disease conditions are very well known, but still there are a lot of work need to to develop new targets because of availability of less medications available in the market,
Exactly.
and
Exactly.
because of, as
Mhm.
you told, the MDR is the main MDR is the main issue with all these relative medication. So yes,
Yes.
thank you so much, ma'am. Thank you, you all. It was very wonderful to have you all with us. We are grateful to that you could have joined us today, and we hope to see you in our upcoming or the forthcoming sessions as well. So yes, tune with us, stay tuned, and let's do it together. Thank you. Thank
Thank you, Dr. Sakshi. You've really conducted the session so well and made
you.
me comfortable, and thank you for that.
Thank you. Thank you, ma'am. Thank you.
Thank you.